Hello everyone, welcome to part 2 on the challenging topics of on the challenging concepts on the special senses topic, or chapter 15 of the Marieb textbook. So we'll continue our conversation with the retina. So on this picture we can see those three cells we mentioned earlier in the last video. I mentioned it's the photoreceptors, the blue and the yellow. The reds are our bipolar cells, and then the ganglion cells will be the gray ones here. So this light actually will travel through, this would be here would be vitreous humor within the posterior segment. It will travel through this way, and then it will be sensed by the outer segment of our photoreceptors. Information will go from photoreceptors to bipolar cells to ganglion cells, and then from the ganglion cells it will go out um, as the optic nerve. Okay, so here is an enlarged view. Now they've just oriented it differently. The vitreous humor and so on will be now to the top of our screen, further up, right? So here are the rods and cones. Rods and cones basically are modified bipolar cells. Here's the cell body, two processes, right? Rods are mainly for dim light and then peripheral vision, while cones are high resolution colored vision, and we'll talk a little bit more about them in the next slide. So non-color vision for rods, because they only have one pigment, so more grayscale, black and white. Cones have color vision, three visual pigments. Rods are highly sensitive to light. What that means is even with a little bit of light, dim light, they will be innervated, they will respond, while cones need a lot of light for them to be able to function. Low acuity for the rods because we have multiple rods that will converge to one ganglion cell, whereas cones, it's 1-to-1. So one cone will communicate with one bipolar cell, which in turn will communicate with one ganglion cell. So each ganglion cell will only receive information from one cone. We have a lot more rods, mainly in the periphery of our retina. Cones, as we get to the center, is where we find it, and it's going to be less in number. The most is in the fovea centralis, which just has um, cones in it. In terms of pigments, I wanted to talk about the retinal. It's part of the rhodopsin. It's found within the rhodopsin. Um, and the retinal is synthesized from vitamin A. Carrots, for example, have a lot of vitamin A. In terms of retinal, there's isomers of retinal. So isomers are same molecular formula, different structural formula. So retinal goes from a bent form, the cis form, to a trans form. So the straight form. This is what happens when light strikes the retinal. They go from a bent form to a straight form. When that happens, so here is an image. So this is rhodopsin, this big blue dark blue purpley molecule, and then the violet colored one is the retinal. This is the bent form. You can see the little hinge there. That's the cis. When light strikes it, it becomes straight. So it will activate a cascade of events, G protein, and then this enzyme, phosphodiesterase enzyme, is very important. That enzyme will break down cyclic GMP. Okay, when cyclic GMP is broken down, of course, the activity of cyclic GMP is lost. Okay, so let's take a step back and just talk about cyclic GMP. So when there is no light, cyclic GMP is present in these photoreceptors. It'll be bound to this channel. This channel is a cationic channel. It's going to allow sodium and calcium to move into the cell. And then recalling what we learned in the action potential, when we have, or even membrane potential, when we have calcium and sodium flowing into our cell, it becomes depolarized. When this cell depolarizes, it will actually release a neurotransmitter from these photoreceptors, and this neurotransmitter is a inhibitory neurotransmitter. So it's an inhibitory neurotransmitter, so it will inhibit our bipolar cells. So when light strikes it, we have broken down cyclic GMP, which means this channel is closed. So when this channel is closed, it does not depolarize. In fact, it actually hyperpolarizes. Okay, so it's hyperpolarizes, so it goes further away from, becomes more negative, right? So when it hyperpolarizes, and please note, hyperpolarization of the photoreceptors is the signal. Okay, is that receptor saying, I have received light. Okay, so when this photoreceptor hyperpolarizes, it does not release that inhibitory neurotransmitter to the bipolar cells. So when the bipolar cells which have not been inhibited now can depolarize, and they themselves now can release a neurotransmitter. This neurotransmitter then goes to the ganglion cell, and it causes the ganglion cell to depolarize. So when the ganglion cell depolarizes, reaches threshold, and action potential is produced in the ganglion cell, it will be transmitted by its axon in the optic nerve, which becomes the optic nerve. Okay, so here's a picture going through that again. So this is our photoreceptor, this is the bipolar cell, and this is our ganglion cell. So in the dark, we have cyclic GMP present. So these channels are open, sodium and calcium move into the cell. This cell becomes depolarized, so it is excitatory, right? It has become it's being stimulated, so to speak. It's going to release a neurotransmitter from the distal end. This neurotransmitter glutamate is actually inhibitory. So when that crosses the cleft and binds to the bipolar cell, it's going to cause hyperpolarization of this bipolar cell. When this bipolar cell is hyperpolarized, it will not release a neurotransmitter itself, so it would not release this. So then there is no information that goes from here to the next cell, so no information. So the ganglion cell will not depolarize and will not create an action potential. No action potential is created within the ganglion cell. Whereas if we look at in the light, so light strikes rhodopsin, retinal changes from the cis to the bent form. That activates a couple of protein, which activates a phosphodiesterase enzyme that breaks down the cyclic GMP. The gate, in the absence of the cyclic GMP, will close. So this cell is no longer becoming depolarized, it actually becomes hyperpolarized. When this is hyperpolarized, it will not release its neurotransmitter. When it does not release its neurotransmitter, now this cell is not inhibited, it can actually depolarize. When this cell depolarizes, it will release a neurotransmitter which will be stimulatory or excitatory to the ganglion cell, and the ganglion cell, once it reaches the threshold, will produce an action potential. Okay, so next slide, we're going to talk about how this moves. So from the ganglion cell, they become the optic nerve. The optic nerve moves to what's a structure called optic chiasma. This is found just above our pituitary glands. At the optic chiasma, it's like an X. Only the medial fibers or middle fibers that are coming from the optic nerve each side will cross over while the lateral fibers stay on. After this point, we call it optic tracts, and optic tracts go to the thalamus, and from the thalamus it will be called optic radiation, and optic radiation will go to our primary visual cortex found in our occipital lobe. Information does travel to other spots, right? So pretectal nucleus are superior colliculi, for example, and so on for reflexes, awakenedness, and so on. But this is the main pathway in which the visual information will go to the primary visual cortex. Next, we're going to do a little overview of the ear. The ear has three major areas: the outer ear, the inner ear, and the middle ear, of course. So we'll talk a little bit about them. So outer ear is basically the auricle or the ear pinna, the external acoustic canal, and then the eardrum, which the scientific name is the tympanic membrane. And then the middle ear is also called the tympanic cavity, air-filled cavity. There are these bones which we'll talk about, ossicles, and then the eustachian tube. And then within the inner ear, we of course have the cochlea, which is part of the hearing, but we also have the semicircular canals and the vestibule and the nerves that take the information to our brain. So in terms of auricle, the ear pinna, it's made up of elastic cartilage. Hopefully you recall from our tissues conversation. That's why it's able to bounce back. The function of the ear pinna is to funnel in sound. External auditory canal. External auditory canal will transmit the sound to the tympanic membrane. It has hair, it has a sebaceous gland as well as a ceruminous gland. The ceruminous gland is what's responsible for the wax, which helps to keep that auditory canal waterproof. Then tympanic membrane, that's the eardrum, which is the boundary between external and middle ear. When sound waves strike this, it will vibrate and then it will transmit those energy to the middle ear, to the ossicles, those little bones. So middle ear is also called the tympanic cavity. It's an air-filled cavity. Let's talk about the eustachian tube, which is also called the pharyngotympanic tube. It communicates between the tympanic membrane and the pharynx. This helps to equalize pressure in the middle ear. So when you go up, say, on a plane or high altitude, you feel that there's a little bit of congestion in your ear, you pop your ears. That's how we want to try and exchange the air to equalize the pressure. You might have heard of otitis media. That's an inflammation of the middle ear. Usually children are at higher susceptibility to this because the eustachian tube is a little more horizontal or less vertical. So the fluid at the back of the throat, the pharynx, can get into the middle ear, and if it remains there with bacteria that's usually present in our pharynx, will cause inflammation and infection here. So a lot of patients, if they suffer with fluid, a lot of children which have fluid in their middle ear, what they will do is grommets or tubes, which helps that fluid not to get collected there, rather flow into our external ear canal. Then the ossicles, these are three smallest bones in our body. Please remember their names in order. So it's MIS, from outermost to innermost. It's malleus, incus, stapes. They will transmit the sound from the tympanic membrane. So malleus is attached to the tympanic membrane. It will transmit the incus to the stapes, and then stapes is attached to the oval window. So it will transmit that sound to the oval window, and oval window is part of the cochlea. Okay, so that's one function of the ossicles, to transmit those sound, but it also a very, very important function. Another extremely important function is that it amplifies the sound. Okay, so people who have lost these bones or they're fallen, it's basically synovial joints that's between these bones, so it's easy for them to fall, and it's not easy to fix them in the same place. So we put artificial prosthesis. We are able to retain this function of the energy being transferred from the tympanic membrane to the oval window, but the ossicles amplification part is not perfectly replicated. So a lot of times these individuals suffer with some hearing loss as well. So next, what I want to talk about is how our inner ear is a labyrinth. So it's like a little maze. So it's called a bony labyrinth. Um, I won't go into a lot of details about it, but this is found in all three of our inner ear components, the vestibule, which is the utricle and saccule, as well as semicircular canals and cochlea. What I would like us to remember is that they are kind of like layered, so sound will be transmitted to the bony labyrinth first, then they will go to the perilymph, which will be then transferred to the membranous labyrinth, and then which will then be transferred to the endolymph to our CSF as well. And this is where we're going to find the receptors. Receptors are going to be actually facing the endolymph liquid, and the vibrations when they are transferred across will be transferred to movement of these fluids, and that vibration is what's sensitized by those receptors. Okay. So let's continue our conversation with equilibrium first. So semicircular canals, it's not half circles, it's really 2/3 circles. There are 3 of them at right angles to each other. Excuse me. You have an anterior semicircular canal, a lateral, and a posterior semicircular canal. They will be able to respond or detect angular motion or rotational movement. So if we did a cartwheel, it's that during that movement, it's semicircular canals that's giving information to our brain in terms of position of our body. Whereas the other part of the equilibrium analysis in receptor containing structures is the vestibule. These are little 2 egg-shaped cavities, the utricle and the saccule. Utricle is closest to our semicircular canal, saccule is a little bit away, US. And these will respond to gravitational changes. So for example, we did a handstand, our head's downwards, right? So it's the utricle and the saccule which will send that information across. You may have heard also some little little crystals have been moved. They're talking about the crystals within the vestibule. Those are the ones that sense those changes. Okay. Next, we're going to talk about the cochlea. Cochlea looks very similar to a snail's shell. It's spiral shaped, conical shaped bony chamber. The receptors itself are called spiral organ of Corti, which basically it can be found at the tip, the apex of the cochlea. Stereocilia, those are the actual cells. So these have what looks like regular cilia, but they're not. These do not move substances across the surface, but they detect movement of substances across the surface. So when fluid flows, they're going to move these cilia, and that they are mechanoreceptors, so they will be sensitized, and they will send the information through to the cochlear branch of the vestibulocochlear nerve. Okay. So in terms of pathway, it's going to travel from our cochlea through what's called the spiral ganglion. Please remember these are bipolar cells. So this being the dendrite, there's going to be an axon, a collection of cell bodies, and then the axon goes. It'll pass through the medulla, goes to our thalamus. Please note, auditory information does go to both areas of the brain, but decide that the brain still gets information more on one side, so they still know which side the sound is coming from. Okay. Next, we're going to talk briefly about the other 2 senses, the sense of smell and sense of taste. Here, both of these are chemoreceptors, chemochemicals, so whether it's chemicals in the air, chemicals in the food that we eat, will be detected by these receptors. Sense of smell is called olfaction, while sense of taste is gustatory. Let's talk about sense of smell. The olfactory epithelium is found on the roof of our nasal cavity. These are basically non-myelinated, which is why you don't get the smell sense immediately, so to speak, within less than a second. It takes a little bit. It's still within a second, but not as quick as, say, sense of vision, for example. And hopefully you recall it's cranial nerve number 1, olfactory nerve. In terms of the pathway, we talk about olfactory tract that takes the information to the olfactory cortex, and some information also goes to our frontal lobe, to the hypothalamus, limbic system, which we need for emotional response to smells. We also need to be able to identify smells, so the frontal lobe's involved in that. Sense of taste, most of our receptors are found on the tongue, um, but we do have some on the soft palate, on the roof of our tongue at the back, on the roof of our mouth. We find in the cheeks, pharynx, as well as epiglottis. In terms of the cranial nerves involved, it's going to be, hopefully you recall, cranial nerve 7, anterior 2/3 of tongue, and posterior 1/3 is going to be cranial nerve 8. These will take the information to our taste buds, from our taste buds to the brainstem, and then to the thalamus, and then they'll go to the gustatory cortex, which is found in a little lobe underneath the temporal lobe. We again send information to the hypothalamus and the limbic system, where we are able to appreciate taste itself. It's the vagus nerve that carries information from the other receptors, whether it's the epiglottis or the lower pharynx. I hope that you found both these videos useful. Thank you for watching and listening. Please have a great day. Have a great day, everyone.